Hello everybody, this is Nolan Bentley, and I am doing a genetics review key. So to start off with, we have this graph up at the top that is showing a seed shape, seed color, in other words, traits. And next to it, we have our dominant symbols for the dominant genes, or also called alleles. So the dominant gene for round seed shapes is capital R, while the recessive allele is lowercase r. Now you'll notice that next to our capital R there is a little blank, and that's because the possible genotypes that would cause a round seed shape only have to contain one capital R, and that's just true for any dominant thing because it doesn't matter what is in that blank spot. It could be a lowercase r or it could be a capital R, and it would still be the same thing. Okay, so that just is applied to all our different questions here. There's also a picture of a pea plant, but that's not actually important. So what I'm going to do here is I'm going to split the screen so that we can just keep that at the top. So I'll split it right there. There we go, now I can just scroll down. So I will be typing up our key as we talk. So starting off, number 1A. It says, what is the dominant allele for flower position? And the dominant allele is axial. That trait, axial. You could also tell me just capital A is fine. The recessive allele for pod shape, as you just come up to pod shape, it is wrinkled, turns out. We know that because it is lowercase. Oh, wrinkled is S. Okay. The phenotype of a capital Y, lowercase y, capital A, lowercase a plant. Phenotype, if you don't know, is the physical I'm overwriting my stuff here. The phenotype is the physical type of something, and that's how I just remember it, pH. But capital Y is dominant, so it wins. So that turns out that Y stands for a yellow seed color. And the capital A stands for placed on the side, which is known as axial. So, and the flowers are placed axially. I'm just going to put axial. I don't know how to properly conjugate that. I'm just going to go check my recording, so give me one sec. Am I recording? Let's pause it. Okay, great. I'm glad that worked. So continuing on, what is the genotype of a heterozygous tall round seed plant? Okay, so first of all, the key here is our heterozygous. That's going to be describing the genotype. So yes, the phenotype, what it physically looks like, is tall. But genotype refers to the actual letters. So for a tall thing that is heterozygous, and hetero means different, we will have to have a capital T and a lowercase t to make it hetero, to make the two genes different. So capital T, little t makes it heterozygous, yet still tall. And a round seed plant could be capital R, either lowercase r or capital R, but we're going to stick with heterozygousness, so lowercase r. Okay, D, or E. What is the genotype for a purebred smooth pod shape short pea plant? Okay, so purebred, smooth, pod shape. So purebred means homozygous, same thing, two of the same gene. So smooth pod, there it is a capital S. So homozygous will be one big smooth to make it smooth, but then another to make it homozygous, or purebred. And a short pea plant, the only way to get a short pea plant is to have a purebred short pea plant. So that will be two lowercase t's. The phenotype of a, should be a, not an, a SSGG plant. Oh, I guess it should be an, starts with that vowel sound. Alright, so the phenotype of an SSGG plant will be two small s's, which makes it wrinkled. Remember, if we ever have two of the same recessive gene, we're able to get that recessive trait. So it will be wrinkled seed shape. Nope, pod shape, apologies. And the little g will make the pod color yellow. That doesn't sound like a very pretty plant. What is the phenotype of capital W, capital A, little r, little r? So capital W refers to the gray seed coat color. So remember, our phenotype is our physical type. So it has gray seed coats. It has capital A. Capital A beats little a because it is dominant. So even though you have axial and terminal genes, only the axial genes are shown or called expressed. So flower position is axial. And our little r, seed shape is wrinkled. Nice recessive trait. Okay. What is the genotype of a short hybrid? So short, let's just start there. Little t. It's got to be homozygous recessive. That's the only way to get the recessive trait. It is hybrid gray seed coat. So it would be W something, but we specified a hybrid, so W little w, to make it a hybrid, and that's it. Remember, genotype is the type of genes, not just the phenotype. Choice I, what is the genotype of a terminal wrinkled seed plant? So terminal will be little a, those are S's. It has to be homozygous recessive, homozygous because it is recessive. So that's how you get a recessive phenotype. It is a wrinkled seed shape, and so it must be little r. R, no capitals. That was weird, yeah. So letter J, what are the two possible genotypes of a green pod plant? Well, the green pod is a dominant trait, so it is G and something, which could be big G, or big G, little g. Either one gives you the same trait, green pea pods, because it's dominant, it wins. Okay, so our anytime we have to do these allele combinations, what you have to think of are four possible gene combinations at max that your genes could separate into whenever the plant makes its gametes or its sex cells. So if you remember, whenever you make your sex cells, you cut your genetic information by half. So this plant, it's in one of its sperm cells or egg cells, whatever it's currently making, it could make a big G, but it couldn't put a second G in that gamete because it's going to pass on only one of each gene. So big G and say maybe big T. Or it could have put big G and little t. But then again, maybe it doesn't put in big G, maybe it puts in little g. Well, if it puts in little g, it has to put in a t, so we can match up little g with big T, or we can match up little g with little t. And that gives us our four different combinations. I like to think of it as first G, first T. Next. First G, second T. Second G, first T. Second G, second T. And that way I get all of our combinations. Now I can try that exact same process with the next problem. First W with the first Y. First W with the second Y. Second W with the first Y. Or second W with the second Y. But here's what you're going to notice is two of those have been repeated, and also the question said what are the two possible? Well, that's because the first two that you got were the same gene, W, little w, and so that limits how many different gametes this plant can make, at least for these genes. And so that's it. It's just these two gametes. If you're putting these on a dihybrid cross, you would put 2 of the first and 2 of the second. That's how you do your dihybrid cross, at least in our traditional way. So of 2 capitals and 2 other capitals where they're both the same thing, that makes it really easy. There is only one type. Big A with big R. Try to find another type. There is none. Good job. Alright, so are we about done with those? Yes, we are. So I'm going to remove this split screen, and we're just going to go on from there. So it says, does this baby... Oh, we weren't done. My bad. Where's my split? There we go. Put that there. Scroll that up, and... There we go. Okay, so the first question asks, does the plant have axial flowers? Now, just for the record, what we're looking at here is like a... see, can I make a ring? It's like a single cell. Oh, that didn't work. Okay, let's not do that. But it's like a single cell. So inside this cell, it has 2 chromosomes. In all of our cells, except for our sex cells and blood cells, we have 2 of each chromosome. Well, inside each of those chromosomes are our genes, and that's why we always talk about them coming in pairs, is because we have 2 copies of each. So in this case, here's this plant's 2 copies. So we want to know if it has axial flowers, and axial is capital A, and the answer is yes, because it has at least 1 capital A. So color pods will be either green or yellow, depending on if it's big G or little g, and it looks like we have G little g, which is the same thing as green pod colors. So it wants to know what homozygous traits we have. Homozygous means 2 of the same gene, so we would be big A, and little r is what it looks like. The heterozygous traits mean they are different, and so in that case it is big T, little t. I always put the capital first, that's just the way I do it. It helps me keep organized. And then big G, little g. Again, I put the capital first. No particular reason, it just keeps things organized and nice and neat. What types of seed shape does the offspring have? So seed shape is R and little r. And it looks like we have 2 little r's, which equals a wrinkled seed. Weird. What is the height of the male plant? Turns out that was a bad question, so I am not asking y'all to do those. I'm not sure how to do my strike through. What is the shortcut for that? Doesn't tell me. Oh well. And also this question doesn't make any sense. This question was originally something slightly different, and I changed it. So I'm just crossing those out, and we'll move on. So just skip all 3 of those. Okay, so now we're at Punnett squares. Alright, and I'm going to have to pause this for a second. Looks like my file is not the right one, so I will be right back. So I'll try to edit out what of that I could. So Punnett squares says read the information below and figure out the following Punnett squares. So it says in a pea plant, T is dominant over short. So as a hybrid tall, and I'm just going to type in what our genotypes are as we go. It's hybrid tall, so that means it is a big T and a little t. And a short plant will have to be little t, little t. Remember, hybrid means one of each kind, not two of the same letters. That's why it was a big and a little. Okay, so then in order to solve our crosses, you arrange one parent on one side of the Punnett square, the other parent on the other. You cross them. I always put the capital on the left side when I cross them. And there we are. We find out, the whatever is in the same column is brought downwards. So capital T, little t, little t. Whatever is on the side is put into the box to its right. So I put this little t with the big T. I put a little t with the first little t. Here I put that little t to the right and the second little t to the right. So I'm not going to repeat that for any more Punnett squares. So it asks, what is the genotype of the hybrid tall plant? Well, it turns out whenever we do a hybrid, it has to be one of each, which makes it by default tall, because the tall gene wins, because it's dominant. So what are the chances of having a tall plant? Well, we look over here and we see that this one is tall, this one is tall, this one is not, this one is not. And thus it must be 2 out of 4, which equals 50%. I could also say half. Half of my offspring given off by these pea plants, I'd expect to be tall. So the gene for albinism, which means no pigmentation, very, very pale, the hair looks almost transparent, the eyes on a true albino tend to be reddish to purple. So a very interesting condition. Can cause a lot of other issues though, so it's not really a very fortunate condition. But it is recessive to normal skin color, so that's good. We're going to let capital A represent normal skin color and lowercase a represent an albino coloring. So it says an albino man. So that will be 2 little a's necessary, because to be albino you would have to receive 2 recessive traits, because if you had a capital it would cover up the albinism. Hard word. A normal woman will have 2 possible genotypes. She could be capital A, or she could be capital A, lowercase a. And so they have an albino child, and that tells you something. If the albino child is little a, which he'd have to be albino, and the father is little a, so I've plugged them in to the side here. Well, in order for this box to have received 2 little a's, the mother must have had a little a. But we do know she was normal, and so we know that her genotype must have been capital A, little a. So that is a lot of logic there, so I hope you can do all that. So it does help to draw your Punnett square, fill it in, and then kind of work backwards. Other than that, I mean, you can just use normal logic. She gave birth to an albino child, and if she was completely homozygous dominant, so 2 capital A's, she couldn't have had that albino child. Her gene would always cover up her husband's. So letter B says, what are the chances that the next child will be albino? Any child born to them, it doesn't really matter what order it's in. Any child born to them has a what looks like a 50% chance, 2 out of 4. Which is the same thing as 50%. So what is the chances that their second child is normal? Again, they don't really affect each other. It's not like you're using up some of these genes. So it is still simply 2 out of 4. You don't just you don't use up these boxes or anything. It's always the same percentage every time they have a child. 50%. So those are those top two that are representing those 2 out of 4 normal children who express a normal phenotype, even though they kind of have an abnormal genotype. So letter D, it's asking about the genotype of their second child, which we just described will be normal. So if you assume it is normal, then the genotype will be capital A, lowercase a. Okay, so those were some tricky questions simply because of that percentage thing. You simply don't use up genotypes. You don't need to do any kind of fancy statistics. Okay, so the gene for curled ears in cats is dominant over the gene for regular ears. We're going to let E represent the dominant allele of curled ears, and lowercase e is going to represent the recessive allele for normal ears. Okay, so curled is dominant. Alright, so let's go through our sentence and make sure we write down anything that we know. A cat breeder crosses a purebred, curled-eared female. Now, we didn't say it's a sex link, so it really doesn't matter that it's female. But it does say purebred, which means 2 of the same gene. So big E, big E. Female, and, like I said, that didn't really matter. And a normal ear male. So normal will be little e, because it has to be little e, and they don't specify homozygous, because the only way to make it normal, so to express the recessive trait, is to have 2 recessive genes. So that's quite easy. All we have to do is plug that into our Punnett square. When you do that, you find out that every single kitten has a copy of the capital E. So there's a 100% chance that it expresses the dominant curled-eared phenotype. So it says there are 5 kittens in the litter. How many purebred curled-eared kittens are there? Well, none. All of them are heterozygous, which is the same thing as hybrids, not purebred. So what genotype would the cat breeder have to cross her female with, which was the 2 capital E, to get all pure breed curl-eared kittens? Well, she would need to breed with 2 capital E's. So one just like her, and that's how you can keep the genes pure. That's why that whole pure breed thing. Okay, so this next one's a little gross. The kittens from this cross mature and breed with each other. So what that means is a cross of big E, little e, and big E, little e. Looks like XXE. Okay. So it asks, what are the chances that there is a purebred curled-eared kitten in the litter? Now I'm going to have to copy a Punnett square to put here. So let's do that. Of course, we're at the end of the page, because that would just make it too easy otherwise. So let me clear this out, and there we are. So big E, little e, big E, little e. We cross those, big E, big E, big E, little e, big E, little e, little e, little e. Okay, so we look at it, pure breed curled-eared kitten in the litter. It's just that one. So that would be a 1 in 4 chance. According to the Punnett square over there. Cool. Alright. So let me clear up some room here so it doesn't take so much room to print off if people need to. And drag that up. Alright, and that just lovely. Alright. So next up is sickle cell anemia, one of the more interesting but fairly awful diseases out there. One of the interesting things about sickle cell anemia is that it is actually a incomplete dominance. And whenever you are heterozygous for it, it actually can protect you from malaria, which is one of the leading, I think it's back to, well, one of the leading diseases in the world, one of the leading transmissible diseases at least. So let's go through this. It says a sickle cell anemia is recessive to circular shaped blood cells. So those are normal shaped blood cells. B is going to represent circular blood, and little b represents sickle cell anemia. So a woman whose mother had sickle cell. Now that's something you'll often see. You'll be like, somebody whose parent had this condition. Because if they had that condition, that must mean that the mother had little b. And so the woman must also have a little b. Now if the woman was sickle cell anemia, we would have let you know. Instead, what we let you know is that she must have inherited this little b from her mother. So she wants to have children with a homozygous normal man. So that's nice. Our homozygous normal man will be, I'm going to put it after the normal because the man's not really important here, will have to be BB, capital B, capital B. So her father was normal, but her brother has sickle cell. Now, that's actually not important to tell us because we already know that her mother had the disease, which meant she must have passed on one of those copies. So that's not even really needed. Not an important fact. At least if we don't talk about the whole incomplete dominance thing. Alright, so if we plug... Oh, I'm sorry. Big B, big B. If we plug those in... big B, big B, big B, little b, big B, big B, big B, little b. The genotype of the woman, based on the fact that her mother had the disease, and we'll assume that her father did not, because her father was normal. Oh, her father wasn't normal. Oh, that's why it says that. Okay, I'm sorry. Back in track. It says her father was normal, but her brother has the disease. Well, that must have meant her father. He must have had it too. Because he must have given a bad copy to her brother. Because if we combine mom and dad's bad copy, we're able to make brother's bad genotype. So, yep, that's what it had to be. Man, it's all in the family. It's rough. So what is the genotype of her mother and brother? Little b, because it's recessive. What is the chance that they will have a child who is a carrier? So the woman and the man, I think they were trying to say. And that is a 50% chance, based off of our chart, which is 2 out of 4. So is it possible for their grandchildren to have sickle cell? I'm assuming they're talking about these people right here. So I'm just going to say, let's just take that out. Now, I get it. I think this question, well, okay, I'll go back and fix it. So I guess what this question was trying to say was, what is the chance that the mother or the original mother and the original father would have a child who's a carrier? So let me copy this. Okay. And we'll paste it here. Nope, that didn't work. Alright, so let me label this one. This is the woman and man this one will have to be the mother and father, also known as the grandparents. Alright, well, I'm not going to bother typing that in. I'll capitalize the father, because fathers are important too. So, if we assume that, let me go back and answer letter D. What are the chances that they will have a child who is a carrier? Well, of the original mother and father, we can say that the father had b, little b, the mother had, how did that get big B, little b? The mother had little b, and so when we cross that, we can say that there's still 50% chance. So that didn't actually change anything, but at least we're matching what they're saying some more. Fortunately, none of the grandchildren look like they will have it. So if we look at the mother and the man and woman's Punnett square, we find that their genotypes do not allow for sickle cell anemia children. So there we go. So C, Punnett squares. Now, if what this question was originally talking about was the grandchildren of the man and woman, so the grandchildren of the man and woman could be sickle cell, because the carriers, the carrier children could find another carrier of the disease to mate with. So that's kind of it. If they found somebody else who had the sickle cell gene, or worse, had sickle cell anemia, that would be the issue. Okay, so let me capitalize that, and we're good. So in pea plants, axial flowers are, oh no, one of these, are dominant over terminal flowers. And there's the second trait. That means you need to do a dihybrid cross. So the gray seed coat color is our other gene. It is dominant over the white seed coat color. So our sentence says a heterozygous axial gray seed coat plant. So let's put all that together. Axial, heterozygous axial will be big A, little a. If we assume that they were also talking heterozygous about the gray seed coat cover, which is what I think they meant, we'll do big G, little g. Oh, and that was slightly too far. That was a little short. Go. Is crossed with a terminal. So terminal will be little a. Has to be two little a's because it is recessive. And the white seed coat must be little g, because it is also recessive. So the first plant was big A, big G, little g. Remember, we can do our different combinations. So the first letter and the first of the other gene, or the first gene and the second of the second gene, or the second of the first genes and the first of the second genes, or finally the second and the second. So each of the 4 combinations. Draw arrows if you need to. You can even make a little mini Punnett square. However, I think it's easiest just to think about the different combinations. So the genotype of the other plant is little a, little g. And this one's easy. If you match the first A with the first G, it's AG. If you match the second A with the second G, it's AG. If you match the first A with the first G, it's AG. If you make the blah 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 blah, it's all AG. AG. So we just do a little A, a little G, and that is the only combination that plant can offer its offspring. Okay, so it's going to start asking for chances now, so we need to go draw our Punnett square. So whenever you draw these Punnett squares, remember it is 4 by 4. So let's plug them in. I'm going to do the easy one first. AG, ag. And here we'll do the other ags. Ag! And just ag. Chill. It's cool. Alright, so now you combine them. So, I'm going to stop that now because I'm starting to annoy myself. But, you can sound it in your own head if you want to while I type. I know you're really bored right now. You could probably just skip ahead. I mean, this really gets kind of boring after a while. Oh, that was fun right there. I almost messed up. It was very exciting. Hey, wait a second. This repeated. I wonder. Let's check it. Little a, little g, big A, big G, big G. Oh my gosh, it's a pattern. Yes, because this plant could only give one thing. It's the same thing all the way down, which saves us so much time. On your paper, you're welcome to just draw an arrow or ditto marks. Honestly, scientists do it all the time if they were doing something like this. Like, let's all be lazy. That's the proper way to approach things. Scientifically, but don't do too much work. So, what are the chances of having a terminal white seed plant? So let's look back at our letters. Terminal is little a. So in order to do that, and let me just, well, no, I'm not going to do that. Little a will be right here, but it's still going to be, nope, that won't be white. So here's the white one. It's two little a's, 2 little g's, 2 little a's, 2 little g's. There we go. That whole column. So that will be 4 out of 16, and hopefully you recognize that as 1/4, or finally 25%. So a terminal gray seed plant. So let's see, terminal is little a, gray seed will be big G, little g in this case. Remember, you only need one capital to get that trait. So that will be the same percentage as our last one, so I'm just going to copy and paste that from now on. And of course it's going to do that each time now. So next up is axial white seed plants. Well, I wonder, axial white seed, hey, it's the same thing again. Huh, another pattern. Who would have thought? And yes, as you may have expected, it's the same thing again. Go check it if you want to. It's true, I'm not lying. Whew, that was good. Let's keep going. Actually, you know what? I'm going to take a break, go save my video just in case everything fails so I don't lose everything. Okay, and we're back. So, number 9, we got round seeds, dominant to wrinkled seeds, yellow seeds, dominant to green seeds. So, again, 2 traits, 2 different traits, 2 different letters. We're going to have to use our dihybrid cross. So, the first thing you have to do is establish what your genes are. So, a heterozygous round. So, round is capital R. It could be capital R and still be round, but then it wouldn't be heterozygous. So we must use a lowercase to make them different, like hetero implies we should. A yellow seed plant, though, could be capital Y, lowercase y, or capital Y, capital Y. They didn't specify, so let's assume they meant to say also heterozygous for the yellow seed. And a wrinkled seed on this other plant means that it is little r, while hybrid yellow means that it is big Y, little y, and actually looking up, I now realize that I accidentally said big Y, little y. So, it says, what is the genotype of the round yellow plant? So, it is big R, big Y, little y. What allele combinations can this plant off to its offspring? That's an interesting way to say it. I think we meant to put give to its offspring. Yep, give. There we are. Let's just go with it then. So, we can give the first R and the first Y. So remember, you had to cut your DNA information in 2 parts. So, that means that one R has to get picked and one Y has to get picked to put into our gamete, which then goes and meets another gamete, which combines with their DNA to make a whole offspring. But the original gamete can only contain one of each. So big R, big Y. Big R, little y is another option. Reread. We got little r, big, oh, that's a D. Big Y, and little r, little y. 4 combinations that the plant can give to its offspring. On the other hand, the genotype of the wrinkled yellow plant is little r, big Y, little y. And its combinations are a little bit simpler. We know that because the first 2 little r's, you don't have any choice. It's just little r. So it could be little r with a big Y, or big Y or little r with little y. That's it. Try to find another combination. You can't, because you have to have one of each type of gene. Okay, so what you got to go do now is plug them into our dihybrid cross in order to determine what your possible genotypes of the offsprings are. So you plug in your gametes first. So big R, big Y, big R, little y, little r, big Y, little r, little y. And down here we got little r, little y. Sorry, little r, big Y. Little r, little y. Hey, we're not done yet. Nope, I got auto-corrected. However, we can just repeat ourselves. Now, it actually turns out that it doesn't change anything if you don't repeat yourself. You could actually just cut out all 4 boxes, leave them blank, and it would not matter. I'm not going to, because I just want to make sure you guys see a consistent version, because the consistent version will just repeat certain rows. So that's what I'm going to do. So now you combine them. Our little R, I always put my capital letter to the left side of whatever group it's in. Big R, big Y, little y, little r, y, r, big Y, little y. Okay. This time we got big R, big Y, little y, big R, y, big R, big Y, y, r, y. Okay, so you may notice that all this should be the same thing as all this. If you're not sure why, try it. You'll find out that I'm correct. So I'm just moving on from there. So let me split the screen so that we can analyze it. Okay. Alright, so what are the chances of having a round, which is capital R, yellow seed plant, and yellow would be capital Y. So round, yellow, not round and yellow, that is wrinkled in yellow. Wrinkled in yellow. Round and yellow, it's got one of each dominant trait. That is round and white. What are they? I'm not sure what the other. Green. That is round and green. That is wrinkled and yellow, wrinkled and green, round and yellow, round and yellow, wrinkled and yellow, wrinkled and yellow, round and yellow. And yes. Cool. So that is 1, 2, 3, 4, 5, 6. So that is a 6 in 16 chance. Now that can be simplified. So 6 in 16, half of 6 is 3, half of 16 is 8, a 3/8 chance. Lovely. Let's not worry about the percentage because that's not a pretty number. So a wrinkled green plant is going to be little r, little y, little y. So here's one, and here's our second. So that is a 2 in 16 chance, which is the same thing as 1 in 8. You may notice that your simplified chances actually match what it would be if you had simplified your Punnett square by leaving out these second 2 rows. But there you go. Alright, so I'm going to remove my split and move on to number 10. So in humans, a widow's peak, which is this little, like, at the top of your hairline, like right where your bangs stop growing, if it comes to a little point pointing towards your nose, it's called a widow's peak. No idea why. I'm sure there's some history why, but we're just going to go with that. I can think of a reason. Probably because it looks like a peak and... yeah, anyways. In humans, a widow's peak, capital W, is dominant over non-widow's peak, little w, and curly hair is dominant to straight hair, at least according to this question. So a non-widow peaked, okay, let's type it in, just to be sure, always write it in, it's easy. So non-widowed peak would have to be lowercase w, a hybrid curly-haired man. So curly hair is capital H something, but in this case we're a hybrid, so it's capital H, lowercase h. A straight hair, so that would be lowercase h, but hybrid for the widow's peak will be capital W, little w. Want to have a child, and apparently they're just incredibly vain and want to know about their children's hair. These people, come on, get your priorities right. Go measure chance of mental defects or something. Something more important here. So the genotype of the child will have to be, the man. We'll put that together is big H, or little h, big W, little w. The genotype of the woman is big H, little w, little w. Of course, you could have flipped around the W's and the H's. There's no proper way to do it. I always keep the capital on the left side, though. So next, we want to know probabilities, which means we need to plug it in to our graph. So what that means is you have to first find your. So we already found the genotypes of our parents, but we also need to find out what combinations they can pass on. So that is your first step. So for the man, he can pass on his little h, and that's all the h's he can pass on. So we can combine that little h with a big W or with a little w. He only gets 2 combinations. His isn't too exciting. The woman, on the other hand, has a big H and a little h, so she combined her big H with a little w. Oh, she's only got one of those. So big H, little w, or little h, little w. So this is a pretty easy cross. So let's do that. Put the man in. Put the woman in. Oh, not wide enough. There we go. Little h, little w. So you can cross those. Let me fix my graph. It's not in the right place. There we go. So you can cross it. H, H, W, W. Man, those W's are just too wide. Okay. Big H, little W, W. Little h, big W, W. Little h, little W, little W. Okay, so you notice that I made it a little bit simpler for myself. So these are it. These are the only four chances. Anything we get from this chart will simplify down to these four. Now, whenever we do these things, we tend to give you 4 boxes, and so we show you the proper way to do it, which we repeated genotypes, but it's just going to be more of the same. Watch, I'll just copy and paste even. And if I actually went through, and looked at these, I would notice that they match up. I was right. It's just repeated. But anyways, that's I guess the full out way. That's showing how I showed you. So I should be consistent. So non-widow's peak will be 2 lowercase w's, with straight hair will be 2 lowercase h's. So according to our chart, we have a 1 in 16, or sorry, 4 in 16 chance, which of course, as I said before, will simplify to 1 in 4, or 25%. Like I said, of those 4, one of them had that trait. Bam, you already had it. So widow peak, curly-haired child. Curly hair will be anything with a capital H. Widow's peak will be anything with a capital W. So if we put them together, widowed peak and curly hair, it has to have a capital W, a capital H, but it doesn't have to have a second one. So this one is, and that's it. So I've highlighted those. It is again a 1 in 4 chance, 4/16. So I'm just going to copy and paste that. Okay. Inserted a tab, but I didn't mean to. Widow peak straight-haired child. So straight hair is 2 little H's. Widowed peak is a W, so it's this one. Again, you'll notice the pattern. It's 1 in 4 again. So there we go. Oh, too much. There we go. And paste. Lovely. Another one. 1. I just went and highlighted those so that you could see that that was still the repeating pattern again. Okay, so number 3. Oh, and thank goodness, then we're done with these dihybrids. Okay, so there are 3 colors of Snapdragon. We have capital R, W, W, and pink, big R, big W. So this is something called incomplete dominance. Dominance. Lovely. So first of all, this is not a dihybrid question. So that's going to confuse everybody. Nobody caught that today. That's going to be confusing. But oh well, that's what happens. That's life. We got it figured out now. Those of you who went to come look at this, you have an advantage. Good. It's healthy. So, it is incomplete dominance. So it says a pink, which would have to be capital R, capital W, and white Snapdragon. So it is white, capital W are crossed. So this is one of those rare cases where the letters will be different. And they're different to show that even though they are for the same trait, the color of the petals of this thing called a Snapdragon, they're different just because they're both dominant. Red and white are dominant. And so whenever they're together, we have this incomplete dominance, and it mixes together to make our pink. Okay, so we are crossing those. To answer our questions, we first have to complete the cross. So there's a pink, another pink, a white, and a white. So the chance of a pink will be 2 out of 4, or 50%. The chance of white will also be 2 out of 4, which is the same thing as 50%. So the next one says a pink and white, pink and pink. That's funny. Pink and a pink. Alright, so let me add a row here. Lovely. Make this all invisible so you can't see it, and you'll just pretend that it's not there. That didn't work. There we go. Okay, I'm happy with that. Alright, cool. So let's fill that in. A pink is RW, and a pink is RW. Didn't mean to italicize that. RW. So you cross those, you get 2 big R's, an R and a W, an R and a W. Oh, that's a T. R and a W, and 2 W's. So that's pretty funny. You cross a pink and a pink, and it produces 3,000 seeds. So it has how many seeds are red? So of these, 1/4 are red. Well, 1/4 of 3,000 is the same thing as 3,000 times 0.25, which is the same thing as 1/4. That calc. That times 0.25, 750 seeds. Of the white ones, we have the same thing. So 1/4 of 3,000 equals the other 750 seeds. And I don't actually have to do this because I could just subtract 1,500, which is the sum of the reds and the whites, but I'm going to do it anyways. So 1/2 of 3,000 equals 3,000 times 0.5, which is the same thing as a half, which equals 1,500 seeds. And if you added them up, it equals 3,000. There you go. Basic mathematics. I hope that helped. So blood type. Blood type's cool. It is something called codominant. In this case, it's got multiple alleles. The chart to the right shows the phenotype and genotype of blood types. That's a lot of typing. So type O. Type O is the absence of A and B markers, unless it's recessive, which just stands for by a lowercase I. So you'll notice we have both 2 codominant traits, A and B, and then we have a normal recessive trait, which is simply covered up by the presence of any dominant trait, either A or B. Okay, so let's go through this. A purebred type B woman. So that means she will have 2 of the same traits. Well, if the trait is B, she has 2 Bs. Oh, okay. Try it one more time. And there we go. A type O man, which means to get the type O, you actually have to get both recessives, just like always, want to have a child. So can they have a type O baby? Let's find out. II, IB, IB. Well, no. Every single child ends up getting IB. So no. Every single child is heterozygous for IB and I. So no. What is the only type they can have? IB. that's not IB. Go. The genotype of their child, I'm just going to copy and paste that. So remember, this would have been a phenotype, so I guess I should say type B. While their genotype is what I was trying to say earlier. So will the child be purebred or hybrid? It is certainly hybrid. It has the information to produce a type O blood, but because that type O blood is recessive, it will not show that trait. Okay, so let me take a little bit of that space out. Type A blood, type A woman. We do not know what she is, so that means she could be homozygous, or she could be heterozygous with an I. And a type O man, going to have to be homozygous recessive, have 2 children. The first child is type A, and their second child is type O. So a type A child and type O would have to be this. However, when you think about it, the only way this could happen, where they're both where the first child gets 2 copies of type A, would be if both parents had type A, which they didn't. So we know that one of the child got 2 eyes, the second child got 2 eyes, which means both parents had to have had an eye. While the other child got type A, which means they must have gotten it from their mom. Too little space. There we go. So if we fill that out, it turns out that the people had a 50% chance of making a baby with type A, heterozygous, and a 50% chance of making a type O baby. So the man must have been II, little I. The woman must have been heterozygous for type A. The chance of producing a third child who is type A, remember, chances, you don't have to worry about the first 2 because every time you have a child, it's its own little independent process. So the first 2 children don't affect your chance of the third child. Remember, these Punnett squares don't get used up or anything. It doesn't work like that. So the chance of their third type is just based off the Punnett square, and thus it is 50%. Same for type O. 50%, it's the other 50%. Okay, cool. Easy problem. Or interesting problem. Alright, so a type AB man. Okay, so that will be... Oop, this one's going to get annoying to type in. And a type A woman. Now, we don't specify if it is a heterozygous or a homozygous woman, so I'm going to say an or, just in case. Haven't finished reading the problem yet, but I am careful. So, those 2 people have a child. The child they bring home has type B blood. So, let's see how that's going to work out. We do know the man for sure, because there's really only one possible way that will work. So type A or type B is what the dad can give. We know the child they have brings home type B blood. So the only way that's going to happen is one of 2 ways. It can either be I capital B with an O, or it could be 2 capital B's. Now, but only one parent has that B type. And thus it has to have the O type attached. That's the only thing that makes sense. So that's how you end up getting your answer. If that child got the B from dad, he must have gotten the I from mom. Which, finally working back, tells you that mom must have been type A with an O. Okay, so we combine those. And there we go. We have our Punnett square. Interesting genotyping. So, can this be their baby? Yes, definitely a hard problem though. What is the genotype of the woman? The genotype of the woman, based off the babies, assuming it's their baby, assuming they didn't accidentally pick the wrong one, it is the woman must have that first genotype with the I. So the probability that they will have an AB child will have to be 1 out of 4, which is the same as 25%. And the probability that they will have a purebred child for A is the same, 1 out of 4, or 25%. Okay. Looks good. Moving on then. So, color blindness. The genotype of a colorblind woman. Oh, more exponents. Great. Has to be X, little h, X little h. So, is hemophilia recessive or dominant? Well, the only way that you get, at least in a female, the disease is if you have 2 copies, and thus it must be recessive. The other way you can tell is that we wrote hemophilia as lowercase, so it is recessive. Now the whole dominant recessive, never mind, we're not going to go there. Now I'm going to have to go back and fix the H's because I forgot it asked for colorblind, so that would be little C's. It's still the same thing. So it is lowercase, and you need, and if you have the capital C, you do not get the disease. That's it. So the phenotype of X little h, X capital H, the big H wins, so that is a normal female. Normal female. No hemophilia. Yay, no hemophilia. So, I'm not going to scroll up. I mean, I think we can remember this. It's pretty easy. So, color blindness, a normal man and woman. A normal man, so it had to be normal. There's no, like, hiding it if you're a man. So, he has to have the capital C and a Y. There's no hiding it because, well, the woman can. The woman is a carrier. So, that means the woman gets 2 X's, but only 1 X with a capital C. And that's C. Ooh, those letters are all too close together. So, they want to have a child. What is the woman's genotype? Well, I just wrote it down, so let me just copy and paste that. She is a carrier. That means she carries the disease, but she does not display it. So it starts right off. We need to go use that Punnett square to determine the chance of children. So I'm just going to copy and paste because, well, I'm kind of tired of typing in my exponents. And Y. I just did that, yep. Alright, so you can fill this out. Go see the beginning if you're not sure how to fill stuff out. And well, they have 1 chance to make a woman like herself, like the mother. and the boy gets a 50% chance, if it's a boy, to be colorblind. So of any children born, though, any child born, it would be 25%. So let's see. Of the chances of a son, within that group sons, what is the chance of being colorblind? Well, it doesn't say that this time. It says, what will the chance they will have a colorblind son? Of any children, it would be 25%. If it has to be a colorblind, but of the group of sons, so let me say, of all children, 50% if you know it's a boy. Actually, no, it's not. Yep, it's just 50%. Because it's got to be 50% because you always know it's going to be 50% of any child. Well, I'll put that. Okay, we're good. What are the chances I'll have a colorblind daughter? Same thing. 25% of all children will be colorblind daughters, while 50% will be if you already know it's a daughter. So if you're ever concerned about that, ask your teacher about whether or not you know the child is a girl or boy when you make the chance. So in reality, that's actually fairly relevant because we can do ultrasounds and actually determine the sex of the baby in the womb. But without doing a genetic test of the baby, we can't determine its genetics for, say, colorblindness. So can they have a colorblind grandchild from their daughter? Well, let's see. If we have two different daughters, it depends. It depends on what daughter it is. So I'm going to say it depends on what daughter. Yes, if the daughter is a carrier, and I just realized that everything I just said was wrong. I'm going to have to edit all that out. Or not, I'll leave it in and show you my mistake. Okay, so what I just realized is that these C's are really close. That big C looks just like that little C, and I thought that was for two little C's. That girl is a carrier. She does not have color blindness. So no. That is a 0% overall. Sorry, my apologies. So the chance they will have a colorblind daughter is for sure 0%. They could have a daughter that carries it, but not that. So if their daughter's a carrier, then they could have a colorblind grandchild. So codominance. Sorry, that was a messy problem. Alright, so codominance is when an organism has two codominant genes, or let me use the proper grammar, two codominant alleles for the same gene. When an organism has two codominant alleles for the same gene, they are both shown. For example, a red and white cow make red and white cattle can combine to make rowan coloring. Rowan coats are spots of red and spots of white on a cow's coat. That is a terrible sentence. Red and white genes. Okay, let me rewrite that. Cattle who have a gene for a red coat and a gene for a white coat can get the trait of a Rowan coat. That is because a Rowan coat is spots of red and spots of white. So there's your example. Incomplete dominance, on the other hand, is when an organism has two incompletely dominant alleles for the same gene. They blend to make a trait in between. And I have remembered that because incomplete, in between, they're similar words. And so it's in between the two traits that make it up. Example: red petal and white petal alleles can mix to make pink colored traits. So notice I didn't say pink colored alleles because there never is one. It is simply if you have both red and white, what it looks like, so the phenotype, maybe I could change traits to phenotype. The phenotype is pink. Overall, it looks pink. Okay. Alright, so our karyotypes, let me zoom out. Alright, so let's read them. So a normal human has a certain number of genes. So let me overview with you. Humans have a total of 23 different chromosomes. However, we have 2 copies of each type. So that would be a total of 1, 2, 3, 4, 5, 6, blah, 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 blah. You'd be getting to 44 here because every time there was 2. So 44, 45, 46. Notice these last 2 are off to the side. I'll get to them. Those first 44 chromosomes are called our autosomes. They are just used for normal, well, they're not just used for normal things, but let me move on. Those are our non-sex chromosomes. They do not determine your sex, what gender you are. While your sex chromosomes are what you inherit that determine your sex, so boy or girl. You get a big ol' X and a second big ol' X if you're a girl. If you're a boy, you get a big X and this little stumpy thing called a Y. That's just, sorry boys, that's all you get. So that's the idea of how we work. We have 44 autosomes, 2 sex chromosomes, adds up to a total of 46 total chromosomes. Notice total. If I ask for pairs, you're going to tell me 23 pairs. Okay, so the idea is to go through here and just kind of analyze what's going on. So I haven't really glanced at the questions yet, so we're gonna look through it real quick. So there's 2 here, 2 here, 2 here, 2 here, 2 here. All of these are pairs of 2. Oh, not right here. 21 is an issue. This person's got an extra 21. That's called trisomy. Anytime you have 3 of the same chromosome, it's trisomy, which is easy to remember because tri means 3, like a tricycle, has 3 wheels. So this person has an extra chromosome, giving them a total of 47 because of an extra one on the 21st pair. So that is called Down syndrome. So karyotype B will have to be Down syndrome. Karyotype D, on the other hand, let's look through it. That's good. That's good. That's good. 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 And this is good depending upon how you feel about boys. But yeah, that's good. That's X and a little Y there. So this person's normal. Looks like. Karyotype C. We got good, 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 good. No! We got an extra X. Alright, so this is something called Klinefelter's syndrome. This person has an extra X, what we call XXY. So, again, 47 total chromosomes, so that's not good. That is a disease, not that bad as far as these type of diseases go. Certainly one of the less awful ones out there. Karyotype A, on the other hand, everything looks good until, again, you get to the sex chromosome. You may be noticing a pattern. The sex chromosome often is the issue. But this time we only have one, and this is a girl. Most of them failed to develop, but if they do develop, honestly, they do okay. It's, again, one of the better types of these chromosomal diseases. But it is called Turner syndrome. Okay, so of the four, which person shows with no... Okay, that's weird. Shows a person without any chromosomal disorders. D. Must be in a boy is a disorder. Just saying. We're a bit weird. So the sex of a person with karyotype B. They have two X's. They're bigger. That's how you can tell they're X's. So that must be a girl. Girls have two X's. The sex of karyotype D is a boy because it's got an X. Boy as an X and a Y. A monosomy is a person that is missing a chromosome in all of their cells. So that's just, not in just any certain cells where maybe the chromosome was removed. That's something that happens. Don't worry about it. Which karyotype is an example of a monosomy disorder? Well, karyotype A is that. Because they are missing the second sex chromosome, whichever one it should have been. A trisomy, easy definition, 3 of the same chromosome. Don't think too hard about that one. The most famous one is Down syndrome. Simply because most people who develop Down syndrome are more people than most other trisomies for Down syndrome survive to birth, and also they're the only autosomal trisomy that develops to adulthood. So, and I believe that's actually Down syndrome, not Downs. So karyotype that shows the trisomy will be the first picture, B. What is the term for the chromosomes that do not provide properly? It is something called non-disjunction. Big fancy word for something that does not. Unjunction? I don't know. I think it kind of makes sense. Non, to not do it. Disjunction, to separate. They don't divide properly. And it's generally the cause of these trisomy disorders. They simply fail to separate during anaphase. Maybe they stick to one side. During either anaphase of meiosis. Okay, so this next section is only if you are a K class. This is looking at the pedigree of Queen Victoria of England, who descended and created a whole bunch of the royal family around Europe. What's pretty cool is she had hemophilia. I guess that's not really cool, but it's interesting to me and other sciencey people. So we're just going to go through this real quick. Here's Victoria. Oh, there's Victoria. She had a bunch of children. Some of them had, like Leopold here, some of them had hemophilia, and thus Victoria must have been the carrier. And that is because the pattern of inheritance is sex-linked. We know it's sex-linked because it seems to only infect the males. It don't affect. It must only affect the males because the boys only have one copy, and so they don't get a chance. Girls like Alice and Irene and Alex and Victoria herself are all able to hide that hemophilic gene because they have a good copy, one from Dad. So, there it is. So I'm supposed to briefly justify my pattern. So we can say this by boys got the disease if they inherited it from one parent, while no girls got the disease. So that's why I chose it. Queen Victoria was the first person that we could trace it back to, although she did not show it herself. So she was a girl. I'm just going to call that one pretty sure. I hope I don't offend any British people out there. I guess I'm British, so at least part of me. So, you know what? God save the Queen. So we got hemophilic. She's good on one, but she's bad on the other. It's not currently working, so there we are. That must have been her genotype. She must have been a carrier. So Leopold was Victoria's only son affected by hemophilia, and you'll notice the only son on here that actually managed to survive to adulthood and make babies. One thing to note is hemophilia caused some pretty rough childhood for a lot of people, and to the point where the average life expectancy, at least when I looked it up, and I'm going to admit it was on Wikipedia, so who knows, but I found a report saying average life expectancy was 11 before modern medicine had changed that. So 11, wow, that's pretty young. I imagine most of them died very, very young, while only a few survived to adulthood, causing a very low overall average. But anyways, got off topic. His genotype must have been X. Oh, not a second X, just a Y. And so if he had the disease on that X, must have been the bad copy of the hemophilia. So currently none of the royal families of Europe show hemophilia. However, the Spanish lineage could still produce the disease. Why is this statement true? Okay, so this one's a little tricky, and what you have to do is you have to go back up here and realize a few things. The main thing is that you have no idea about many of these women. For example, Victoria here never produced a child with hemophilia, but that actually doesn't mean she didn't have it. She did produce a boy, and that means she gave one of her X's to the boy while the dad gave a Y. And so that's a great way to see if somebody has a sex-linked disease. However, the boy didn't show it, which means the Victoria must have given him a good copy. But that doesn't mean she didn't have a bad copy. However, Wilhelm, I think that's what it says, maybe it's just William, I can't really tell, it's too zoomed in, apparently never made any children, or at least never any children with hemophilia. So, well, obviously not. Sophie of Greece never made any children either. So, we don't know about Victoria. She is a maybe. She maybe was a carrier. We'll never know. And that's kind of the tricky thing about these graphs, is they simply can't know some things without, digging up their bodies and genetically testing them. So we just don't know for this one. We don't know for this one. We don't know for this one. And where's our Spanish royal family? Our Spanish royal family's over here. So let's see. Beatrice had it because she gave birth to another Leopold and a Maurice. And I can't tell. I'm going to guess Eugenia. Eugenia. It's a good name. So Beatrice gave birth to Eugenia, apparently was also a carrier because she also had hemophilic boys. So here's where it gets iffy. Here we have the modern Spanish royal family, apparently. And they're descended from whoever this is. Well, whoever this is still hasn't never produced or still hasn't produced any hemophilic children. But that doesn't mean she's not a carrier. We may just don't know. Because any of these girls could just be hiding it. If their father doesn't have the disease, which he doesn't, or we'd have to show it, and their mother is a carrier, one of or two of them, or three, or all of them may have received a bad copy from mom. So we'll never know until somebody tests them or they give birth to a hemophilic male. So that is why it's true. They are descended from people who could have been carriers. And I'm going to call it at that. And that is the end of this review. I sure hope it recorded everything. I hope you have a great day. Good luck, everybody. I wish you the greatest of luck. Thank you. Have a good day.